What are the 4 types of large biological molecules?
Lipids, proteins, carbohydrates, and there's one more. Lipids, proteins, carbohydrates, polymers.
You tell me. Are polymers a 4th type of large biological molecule? What is a polymer?
Polymer is just a large molecule of smaller molecules of monomers.
Yeah, so do you see that's not really a type of large biological molecule. It's just a characteristic that some large molecules have. Okay, so it looks like perhaps you're not remembering the 4th one?
Is it nucleic acid?
That's it, yes. All right, so that question I just asked you, not many classes can be summarized in one question, but that kind of summarizes your whole biochemistry course. So that's a good thing for us to review. The most important question in biochemistry is, what are the 4 main types of large biological molecules? Obviously, you'll need to know a lot more than that to get a good score in the course, but that's the first thing we need to know. So to review, what are the 4 types of large biological molecules?
Hold on.
Yeah, take your time.
One is proteins. 2 is carbohydrates. 3 is lipids. 4 is nucleic acids.
Okay, that's right. What are the 2 main types? What are the 2 types of nucleic acids?
DNA and RNA.
That's right. So that was the one you were having a little trouble remembering. But obviously, those are important molecules in the body. I'm sure you studied those in your biology class. What we've been focusing on are proteins. Are proteins polymers?
Yes.
Because they're made up of repeating subunits. What's another name for a protein polymer?
Amino acid chain.
Okay, I guess you could call it an amino acid chain. What's another name, though?
Polypeptides.
That's the one I was going for, yes. And if it's shorter, it might just be called a peptide. And if it's longer, it might just be called a protein. What's the name of a protein monomer?
Amino acid.
So these are some of the most important questions about proteins. Good. Okay. Were you able to memorize the 20 amino acids?
Actually almost there. I'm in the process of memorizing all of them.
Okay. All right. So that's important. That's good. That's important task to finish because remember, this coming week, your instructor is going to be on to a new chapter with a bunch of new things to memorize. So we need to get that down. Okay. Well, why don't you turn on your screen? All right. Have you memorized the structure of valine?
1 second. Yes.
Let's draw valine. Draw the entire amino acid, please, for valine.
Okay.
Okay. All right. Let's write down the 3-letter abbreviation for valine. Good. Put a squiggle between the main chain and the side chain. Good. Put an alpha on the alpha carbon. Excellent. Okay. That is correct. Incidentally, looks like you already have this memorized, but the common memory aid here is, do you notice that the side chain kind of looks like a V, or this way it looks like an upside down V?
Yeah.
So that's a memory aid that's sometimes used for memorizing the structure of valine, since it starts with, the word starts with a V. Okay, good. Very good. You got that down. I think you said you don't need to know the 1-letter abbreviations?
Right.
Okay. So, what is physiological pH?
7.4.
Good. So, write down what would be the forms of the carboxy end and the nitrogen end at physiological pH. You can just include that in the same picture.
Say that again.
Remember that the carboxy end and the amine end have 2 different forms. Remember we discussed that last time because they're acidic or basic?
Right.
So write down whether they should be in their protonated or deprotonated forms. You can just draw that in the picture you already have. Draw the carboxy end and the nitrogen end at the form they would take at physiological pH. Do you remember one of the things we did in the last session was learn how to draw the correct form, whether it's protonated or deprotonated at different pHs. Do you remember how we would do that?
What do we need the pKas?
That's right. But remember we talked about memorizing the ballpark pKas? Do you have these ballpark pKas memorized?
Oh yeah. pKa starts with R9.
There you go.
Which is around 2.
Yes. However, you shouldn't write it as an OH because that would be an alcohol, and alcohols have a very different pH. What type of what type of functional group do we have there?
That is hydroxy.
It is a hydroxy group, but that's not the most important aspect of it. There's another name for that functional group that involves the carbon that's double bonded to the oxygen.
Say that again. Sorry.
So what type of functional group? Point to the carbonyl carbon. What type of functional group is that in? What type of functional group is that?
That is a carboxylic acid.
Yes. So that's how you should write it over off to the side with the COOH is my point. Is it an alcohol? No. Just not all OHs are considered alcohols. That actually turns out to be quite important in the course. This is not an alcohol group. It's much more acidic. It's a carboxylic acid group. Okay, so that's a better way to write it. Okay, so now let's continue and decide whether each of those should be in their protonated or deprotonated forms.
To become since the pH is 7.4, this is more acidic, so this is going to end up donating protons here. This is going to be NH3 plus. This is going to end up. Can I draw arrows? That be more useful.
If it works for you, sure.
So is this right in the human body?
So is that your guess that the way you have it written would be at physiological pH?
Yeah.
Okay, good. Yeah. So you, as it turned out, you'd already written it that way, but we wanted to confirm why that was correct. Okay, that's right. So that came out to be correct. All right, that's the type of question you're likely to see on the exam, but that's the right way to do that. Let's see. Okay, let's do another example of that then. Let's do pH of 11. Let's redraw the molecule. You can write it at a different place on your screen. Let's draw what the molecule would look like at a pH of 11.
You want me to redraw the molecule, you said?
Yeah, you'll need to do that to answer the question. So draw what the molecule, instead of just modifying the previous picture. So we can start by writing down we're doing a pH of 11.
Okay.
Very good. What is the net charge on the molecule at a pH of 11?
Negative.
Let's be more specific. -1, -2, -3.
-1.
Okay, good. So that's important. I guess we should have done that in the previous example too. So let's scroll back up and figure out the net charge at physiological pH.
This would be just neutral.
Good.
Is that this sign?
I'm not quite sure what you're trying to draw, but no, you could just say it's just 0. The charge is 0. It's just a 0. If you want to show that it's a 0 and not the letter O, you can draw it as a 0 with a slash going from southwest to northeast. And extending a little bit outside of the, yeah, that's just, that's not a biochemistry symbol, but just in mathematics. Mathematicians use that to show that they're talking about a 0 and not an O. So then there's no special symbol for neutral, but it has 0 charge. So we just write down as 0. That's a good way to write it so you don't confuse it with oxygen, say. Okay, good. We should write down that's the net charge, because obviously there are charges inside the molecule. That's just the net charge. Now, your textbook has a good way of classifying these molecules. It says that side chains fall into 4 different types. The 4 types of side chains are nonpolar, polar, positively charged, and negatively charged. I think that's a good way to think about it. So to summarize, the 4 types of side chains are nonpolar, polar, negatively charged, and positively charged. So that would be good to have in your notes. You can tell me when you have that down.
Polar, nonpolar, negatively charged, and what?
And Well, you tell me. What would be a logical last category?
Positively charged.
That's right. Do you see how that basically covers the only possibilities? Those are very logical categories. Those cover the only possibilities.
Okay.
Okay, so which type of side chain does valine have?
Valine has a polar. Side chain is non-polar.
I'm sorry, what was your answer? What type of side chain does it have?
Oh, it has a non-polar side chain
Okay, how do you know?
because there is no charges.
It's true there's no charges, but that just means that doesn't tell you whether it's polar or non-polar. So the way that that's from an earlier chapter that we haven't had a chance to talk about here yet. You determine whether the bonds are polar by asking if there's an electronegativity difference between the atoms. Maybe this is a review of some stuff we talked about in organic chemistry. So usually, who's more electronegative in the periodic table? Things that are further to the right or further to the left?
To the right.
Yeah. So usually things that are further to the right are more electronegative than things that are to the left. But do you remember we talked about in OChem, that doesn't work for hydrogen. Hydrogen's electronegativity is not given by the periodic table. We just need to memorize that the electronegativity of hydrogen is similar to another element. Do you remember talking about that in OChem? Do you know what other element has similar electronegativity to hydrogen?
Say that again.
What other element has similar electronegativity to hydrogen? Because
Lithium.
so it looks like you're maybe trying to use the periodic table for that. You might be saying that because they're close to each other in the periodic table. But the point I was making is that for most elements, you can estimate the electronegativity using the periodic table. But that doesn't work for hydrogen. You cannot estimate the electronegativity of hydrogen using the periodic table. The periodic table does not give us a good indication of the electronegativity of hydrogen. So instead, we just kind of have to memorize. Well, what we saw in OChem is that the electronegativity of hydrogen is similar to carbon. Notice they're not next to each other in the periodic table, so that just has to be memorized. The electronegativity of hydrogen is similar to carbon. The electronegativity of hydrogen is similar to carbon. That's a very important idea, so you can tell me when you have that in your notes.
Okay.
So notice that in your side chain, the only bonds are carbon-hydrogen bonds. In your side chain, the only bonds are carbon-hydrogen bonds. Do carbon and hydrogen have similar or different electronegativity?
Carbon has similar electronegativity.
That's right. Does that mean they would have a polar or a non-polar bond?
Non-polar.
That's right. Do you remember what does non-polar bond mean? And what does polar bond mean?
Polar bond means it has a slightly negative charge and a slightly positive charge.
Okay, that's right. A more fundamental definition is a polar bond has an unequal sharing of electrons, and a non-polar bond has equal sharing of electrons. That would be good to have down in your notes as well. A non-polar covalent bond has equal sharing of electrons, and a polar covalent bond has unequal sharing of electrons. So you can tell me when you have that in your notes.
Okay.
Okay. All right, so what did we decide? Would this side chain have polar or non-polar bonds?
Non-polar bonds.
So your answer that you gave before was correct. You ended up saying this was a non-polar side chain, and you were correct. But it's important to understand why. By the way, obviously the main chain is not non-polar. The main chain is polar and has charges. But the side chain here is non-polar. Okay. Now, you and I don't think we've talked much about hydrophobic and hydrophilic terms, but those are terms that your instructors talked about. So would you say this side chain is hydrophobic or hydrophilic?
Hydrophobic.
And how do you, what's your logic there?
Because water is a polar molecule and like dissolves like.
That's a good answer. Yes, that's a good answer. So non-polar things don't dissolve well in water. Do polar things dissolve well in water?
Yes.
Do charged things dissolve well in water?
Yes.
Yes. So things that are polar and charged dissolve well in water. In fact, who dissolves better in water? Something that's polar or something that's charged?
Something that's charged.
Because it just has bigger charges to form attractions with the water. Okay. But something that's nonpolar is not going to dissolve well in water. So this is a fairly hydrophobic side chain. That turns out to be important in biochem. Okay, so that's another important idea.
Give me 1 second.
Do you remember the idea of a stereocenter from organic chemistry?
Yes.
Point to the alpha carbon in valine.
Okay. The alpha carbon.
Is that a stereocenter?
Yes, that is.
How can you see that?
It's attached to 4 different molecules.
That's right. So the picture that you've drawn here, it looks like you memorized a page that was a bunch of, these are basically Fisher projections. We're supposed to assume that the horizontal bonds are pointing towards you out of the page, and the vertical bonds are pointing away from you into the page. And when you do that, you would get the correct way to write this stereocenter is this. There's 2 different names for the positions the stereocenter can take in a protein. Now, in organic chemistry, we learned that in general, stereocenters can be either R or S. But for proteins, there's 2 different terms. Did you pick up from class? What are the 2 different terms that are used for a stereocenter? What are the 2 different terms that are used for the stereocenter in a protein? For proteins, we don't use R and S. As your instructor mentioned, do you remember what are the terms that are used for proteins?
I don't remember.
So those would be L or D. L or D. And the convention is that they're called L and D. Well, naturally occurring proteins are all L. Naturally occurring proteins are all in the L form, which actually turns out to be the S form. So if you actually used your organic chemistry skills here, you would see that you had drawn this Fisher projection in the S form. I didn't see too much about that in the notes you sent me. That's in your textbook. Most classes cover that. Maybe your instructor didn't go into that in too much detail, though. So, okay. Let's see. What's the name of this amino acid?
That amino acid is valine?
Yes. This is just another picture of the amino acid we were just talking about. I think it's important to know how to draw the amino acids in this structure as well. The Fisher diagrams that you learned are good, but they can't be linked up with each other. Remember, the whole point of learning about amino acids is that shortly we're going to learn how they can link up into a peptide. But you can't link up those Fisher diagrams. So there's nothing wrong with knowing those Fisher diagrams, but we also need to learn how to write these like this. So I would encourage you from now on, if you're learning the Fisher diagrams, to also make sure you know how to draw the diagrams like this. If you want to show the stereocenter here, you could do this with a wedge, but I'm not sure if your instructor cares about this too much. So the stereochemistry here, in order to make this S, you'd have to put this on a wedge. And then suppose that we flipped it. Suppose I took this molecule and I flipped it 180 degrees around this axis. Would this group now be on a wedge or a dash down here?
Dash.
So the stereochemistry here. So my point here is it's usually best, it turns out to be best to draw amino acids with the N on the left and the carboxy on the right. the best way to learn how to draw these for most purposes is to put the amino group on the left and the carboxy on the right. When you draw it in this orientation, there's a wedge. When you draw it in this orientation, there's a dash. Although maybe your instructor, I didn't see too much about stereochemistry in your notes, so maybe your instructor would let you just draw it as a solid line. In fact, to keep things simple, maybe that's how I'll draw it now. We won't worry too much about the stereochemistry, and we'll just draw it here as a solid line. So these would be two ways of drawing this structure here then. The reason why this is better than the Fisher diagrams is because this is the way that we can link one amino acid to another amino acid. You can't link Fisher diagrams together. So like I said, there's nothing wrong with the way you just drew it. I think maybe that's one of how they were drawn in the handout your instructor gave you. So that's a perfectly good way to memorize them. But you also need to know how to draw them this way. So the Fisher diagrams are fine, but you also need to know how to draw them this way. What's the name of this carbon?
Alpha carbon.
Yeah, so notice it's nitrogen, alpha carbon, carboxy carbon. Nitrogen, alpha carbon, carboxy carbon. It's usually best to draw the nitrogen on the left and the carboxy on the right. Nitrogen on the left, carboxy on the right. That's the conventional way that these are usually drawn. All right, let's see. Okay, so let's turn your screen on again, please. Let's go to your drawing program. All right, good. So now let's draw serine. And obviously we want to do this from memory. We're trying to practice having memorized these, so let's try drawing serine. And good.
Do you want me to draw it in the way you said before, like where the nitrogen on the left and the carboxy on the right?
It looks like you memorized it in the Fisher diagram. So let's stick with that, but draw it both ways. So let's draw it the way I just was mentioning as well. Yes, because that new way I was talking about is going to be necessary as we go on.
Okay.
Okay, excellent. Now let's draw that picture as it would look at physiological pH. Modify that picture so it looks like what it would look like at physiological pH.
No. Oops. So this would be good?
Yes, it's already good. All right, very good. So do you remember we were talking about how there's 4 types of side chains? What did we say were the 4 types of side chains?
They are polar, nonpolar, negatively charged, and positively charged.
Which type is this in serine?
In serine, that is polar
All right, how do you know?
because of the alcohol.
Why does that make it polar?
Because they are, it's unequal sharing of electrons.
Good, that's right. How do you know they have unequal sharing of electrons?
Because oxygen is extremely electronegative.
That's right. How electronegative is hydrogen? How do you judge the electronegativity of hydrogen?
It's very weak because it's all the way to the left.
You mean it's in the far left column of the periodic table?
Yes.
Okay, but do you remember how earlier in the session we were talking about how the periodic table is not helpful for hydrogen?
And, its electronegativity is similar to carbon.
That's right. So what was wrong with what you just said? What you just said made it sound like hydrogen is less electronegative than carbon because it's further to the left than carbon, but that actually turns out to be mistaken. Hydrogen is similar to carbon. So what we need to memorize about hydrogen is that we should not use the periodic table when we're judging the electronegativity of hydrogen. Instead, we should just memorize that hydrogen is similar to carbon in electronegativity. But carbon is to the left of oxygen. So as you were saying, it's a polar bond between the oxygen and the hydrogen. What would be the delta charges there? What would be the delta charge on the oxygen and on the hydrogen?
Oxygen would be delta negative and hydrogen would be delta positive.
That's right, because the oxygen is further to the right. Okay, that's correct. So would this tend to be more a hydrophobic or hydrophilic side chain?
Hydrophilic.
Because it's polar and like dissolves like. Okay, that's good. All right, so that's a good picture here for serine. Very good. So, one moment. Okay, that's good. So now let's try drawing tyrosine.
Tyrosine, you said?
Yes.
Okay.
Okay, that's very good. That's correct. Okay, good. So does tyrosine have any groups that could have two different forms?
Yes, the nitrogen group and the carboxy group.
Okay, that turns out to be only a partial answer because in tyrosine, the side chain can also take two forms. That's something we should memorize. The side chain of tyrosine can also take two different forms. The side chain of tyrosine can also take two different forms. So what's the pKa for the nitrogen?
Hold on, let me write this down.
Sure.
So you said tyrosine can take on two different forms?
The side chain can.
Okay. Say that again.
What's the pKa for the nitrogen?
pKa for nitrogen is around 9.
Good. And the carboxy?
The carboxy is around 2.
Good. Do you have your textbook with you?
Yes.
Turn to page 41, please.
Do you have the same book?
Hopefully. Do you have table 3.1 on page 41?
Page 41. Yes.
Excellent. All right, so this is a real useful table here. Do you see tyrosine there?
Yes.
Okay. Do you see the two different forms of tyrosine?
Yes.
All right, and what's its pKa?
10.9.
Good. Now, we only need to know ballparks, which I think basically means integers. So you just, basically, I think you need to memorize that table, but you should just round all the pKs off to the nearest integer. So I think when your instructor said that they want you to know the ballpark pKa's, they meant that they want you to know this table, but you don't need to know the decimal points. You can just round it off to the nearest integer. So what should we memorize as the pKa for the tyrosine side chain?
Sorry. So we should know it's around 10.9, so 11.
That's right. So you don't need to memorize 10.9. You can just memorize it as 11. I think that's what they mean. That's what most courses mean by ballpark.
Mm-hmm.
Okay. So basically, notice this table tells you which side chains have more than one form. This table tells you which side chains have more than one form. So you kind of have to memorize this table. It tells you which side chains have more than one form, and it tells you what those forms are. So this has to be memorized in most courses. I think your instructor said they wanted you to know the ballpark numbers, so I think they meant memorize this table. That's pretty typical. Okay, so let's say that we have a pH of 12. Let's write down what tyrosine would look like at a pH of 12. I changed my mind. Let's write down what tyrosine would look like at a pH of 10. Let's write down what tyrosine would look like at a pH of 10.
Let me write this down on the side. That's better. So OH should be around 11. Correct?
Yes,
pKa.
that's from the table, right?
Yeah.
Yeah, so that's, like I say, a number you should memorize. Good.
Okay, so this would be.
Oh, wait. Okay, yeah. So that's, I'm sorry, that's fine. Okay, keep going.
So would this be positive?
Off to the side, draw what are the two different forms?
Oh, no, it would just stay like this.
Good.
The two different forms would be.
But for practice, let's draw the two different forms off to the side. How do you know those are the two forms? Because they're in the table. So that is a part of the table that has to be memorized. That's definitely important. We need to memorize what the two forms are for each of those side chains. Okay, so tell me when you're done drawing tyrosine at a pH of 10.
I'm done.
Excellent. All right, so notice you couldn't do that if you hadn't memorized that the ballpark side chain pKa was 11, and if you hadn't memorized what the two forms look like. So good news that there's more work for you. So that's some extra work for that chapter. Now let's draw a separate picture. Let's draw tyrosine at physiological pH.
All right.
Excellent. So what's the net charge at physiological pH?
Neutral.
Good. What was the net charge at pH of 10?
-1.
Good. Let's do one more. Let's draw tyrosine at a pH of 12. pH of 12.
All right.
Very good. So what's the net charge at a pH of 12?
-2.
-2. So notice you can't just say negative. You have to say -1 or -2 or -3. So which picture shows tyrosine at physiological pH?
At physiological, the middle picture, neutral.
Good. So again, which type of side chain is this? Polar, nonpolar, positively charged, or negatively charged?
Polar.
Right. But now notice in the bottom picture, the side chain was charged, right?
Mhm.
But it's not, we don't put it in the charged category because the categories are based on what these look like in the body at physiological pH. The categories are based on what the side chains look like in the body at physiological pH. At a pH of 12, the side chain would be negatively charged, but this is not considered a negatively charged side chain because it doesn't look like that at physiological pH. Does that make any sense?
Yes.
Okay. Okay. Very good. So let's now try aspartic acid. Try drawing aspartic acid at physiological pH. Aspartic acid at physiological pH.
Aspartic acid. Is that the same as aspartate?
Oh, good point. So that's basically the same thing as aspartate. That's right. So that's right. Your handout might have called it aspartate. That's fine. We'll do it at physiological pH.
Okay.
Okay, so what's the net charge?
The net charge is -1.
Very good. Okay, so let's look at that side chain here. Does the side chain have one form or two forms?
That side chain can have two forms.
Because what type of functional group is there on the side chain?
Carboxylic acid.
Yes. So here, point to the side chain carboxylic acid. Point to the main chain carboxylic acid. So it's important to use those terms so you know which one you're talking about. What do you think is the ballpark pKa for the side chain?
The ballpark pKa was probably around 2.
That's not a bad guess, but check that table in your textbook.
.1.
So we can round that. Sorry, what were you saying?
So it's around 4.
Yeah. Now I'm not sure how much of a stickler your instructor is going to be for this or not. Whether you need to know that it's 4, whether you can just keep using the 2. You could ask them whether they care about that. But it turns out it's slightly different than the main chain. The pKa of the side chain is slightly different than the main chain because your environment can change your pKa. Many courses would require you to memorize that it's a little bit different than the main chain. I don't know whether your instructor is into that or not. But anyway, you can see the safest thing is just to memorize the table. It's actually at 4. Does that change your answer?
No.
Okay, so we still got the same picture here. So which of the 4 types, so that's good, that's the side chain, good. So which of the 4 types of side chain do we have here?
Which of the 4 types of side chain? We have a polar molecule side chain.
Do you remember what are the 4 types? What are the 4 types?
Polar, nonpolar, negatively charged, positively charged, so it would be negatively charged.
Yes. So we don't want to, it's not just polar, it's negatively charged. So it would go in the negatively charged group at physiological pH. Good. Okay. At physiological pH. Good. Do you see now why this is sometimes called aspartate and sometimes called aspartic acid? Why does it have those 2 different names?
Because depending on the pH, it'll take on an acid form or the negatively charged side chain form.
Yeah, so which form do you have drawn here? The aspartate form or the aspartic acid form?
The aspartate.
Yeah, but if we change the pH, it would have the protonated form. Actually, oftentimes people use both terms for both forms. So sometimes people will just call this a deprotonated aspartic acid. So anyway, you need to be, after all, you need to be comfortable with the idea that both names are correct. Both names are correct. After all, what name did they use in table 3.1?
I think they use aspartic acid.
Yeah. On the other hand, in that handout, you saw it was called aspartate. So both of the, you need to be familiar with both of those names, is my point. Okay. So anyway, that is the correct picture there. Okay, good. Have you memorized arginine?
Yes.
Let's draw that.
You said arginine, right?
Yes. And let's draw it at physiological pH.
This is what's going to do. Okay, that has a +3 configuration. Charge.
Okay. All right, net charge. That's good that you were thinking about that net charge. So how many different forms does the side chain have here? Does the side chain have one form or two forms?
2 forms.
Write the other form off to the side.
What do we like? That.
Okay. 1 second. Let's see. Okay. So I was just telling you that you can memorize the 2 different forms of the amino acid side chains from that table 3.1, but I actually don't really care for the way they drew arginine. So why don't you actually memorize it like this instead? Maybe you can write it next to the table. I think this is a better way to write it. So these are the 2 forms. These are the 2 forms of the arginine side chain. You can tell me when you have that down.
Okay.
Okay, these are just the side chains. So do you see what you had on your screen was incorrect, right?
Yes.
Because you had 2 charges here. So it turns out only one of the nitrogens has 2 forms. Only one of the nitrogens has 2 forms. I think you were misunderstanding what the textbook was saying. Basically, this nitrogen has 2 different forms. This nitrogen has 2 different forms. This is a basic nitrogen that can gain a proton. This nitrogen doesn't change form. Okay, I think these are probably the best 2 ways to memorize the 2 different forms of the arginine side chain. Do you see how they have it written in table 3.1 in your textbook?
Mm-hmm.
Notice that they're using those dashes. So what they're trying to do is they're actually trying to say that there's another resonance structure. If you remember resonance from organic chemistry, There's another resonance structure where the double bond is over here and the positive charge is on this nitrogen. So they're trying to be a little bit fancy and combine both resonance structures into one picture. So the picture in table 3.1 is trying to show that there's two different resonance structures. One resonance structure where the double bond is here, and one resonance structure where the double bond is here. So they use some dashed lines to try to show that. But I think that that's probably unnecessarily complicated for your course. I don't think we need to worry about that resonance for your course. For your course, I think it would be better to just learn them in these two forms. I think that'll be less confusing. So I would maybe make a note, add that as a note next to the table in your textbook. This is a better way to memorize arginine. Okay, let's see. So the key thing is it's the double bonded carbon that's gaining or losing a proton, not this single bond. I'm sorry, it's the double bonded nitrogen that's gaining or losing a proton, not this nitrogen off to the side when we draw it in this form. All right. There's no time like the present, so let's try to memorize that. Stare at this for a while and try to memorize these two side chains for arginine. Tell me when you feel you have that memorized.
I think I have memorized.
Very good. So let's turn on your screen. Okay. All right, so let's continue to draw arginine at physiological pH. If you want to, you can just fix the previous picture. So that previous picture was incorrect. So let's go back and fix the previous picture so it shows arginine at physiological pH. Let's try to do that from memory without looking at your notes.
So this would be okay.
So notice that that was important because it changed what the charge was. Remember, what was the most important factor in organic chemistry? The charges. So the charges are very important in biochemistry as well. All right, so now I think that we've fixed that. And off to the side, you were trying to write the other form. I guess you can erase what you have written off there on the right because that turned out to be incorrect. Or maybe, yeah, that's kind of incorrect off to the right. So you can erase that. All right, I was mentioning that that fragment you have written off to the right turned out to be incorrect. So you can erase that on the right side of your screen, the other form of the side chain. All right. Now, actually, we're not done. You actually have no way of figuring this out yet because I don't know if you know what's the ballpark pKa for the arginine side chain. Notice that the arginine is not really an amine. The arginine side chain is not an amine, so you really can't assume that its pKa is 9. You probably don't have that memorized yet, so go ahead and look it up in the table 3.1.
12.5.
Yeah, so I think you should just memorize that as 12.5. It's hard to round that off, so let's just memorize that as 12.5. That's the biggest pKa in the table. So the side chain here has a pKa of 12.5. That should be memorized. So is your picture correct or incorrect at physiological pH?
It's correct.
Okay, good. All right, very good. So now let's try a pH of 11. Draw arginine at a pH of 11.
So this would be. Oh wait.
let's actually redraw the whole picture so that we can keep this in your notes.
Oh. Okay.
So what's the net charge?
Net charge is neutral.
Excellent. That's very good. Let's also draw the arginine at a pH of 13.
It's a different picture.
Yes, please. pH of 13.
Everything's done in protons. Okay.
Okay, so what's the net charge?
-1.
Good. So point to the nitrogen at the end of the side chain. Or actually, I should say, how many bonds? So yeah, point to the nitrogen in the northeast corner.
This one.
Yes. How many bonds should a neutral nitrogen have?
A neutral nitrogen should have each 3 bonds.
And what's the formal charge on that nitrogen?
Neutral.
So does it have the right number of bonds?
Yes.
Yeah. How about check the other nitrogens? Do they have the right numbers of bonds?
Yes.
All right, so that's one way you can check that your picture is coming out correct. Neutral nitrogens have 3 bonds. Nitrogens with positive charges have 4 bonds. That's a good way to check that your pictures are correct. Neutral nitrogens have 3 bonds. Positive nitrogens have 4 bonds. That's a way that we can check. Okay, let's go back then to the physiological picture.
The physiological picture.
The picture at physiological pH. So which of the 4 types of side chain does arginine have?
Arginine has a positively charged side chain.
At physiological pH. Good. By the way, I don't know if your instructor cares about this. cares about spelling, but arginine is spelled with 2 I's and no E. If you look at, you wrote down the word there, so that's 2 I's, no E. And what's the 3-letter abbreviation?
ARG.
Okay. Okay, so we learned some important stuff. Arginine is one of the amino acids that's most likely, I think, to come up on the next test because students have a little trouble with it. So this is an important one to make sure you have down. This is pretty sure to be on the test. So let's give ourselves some space and let's draw histidine. Let's try drawing histidine at physiological pH.
Okay. All right.
Okay. So does the histidine side chain have one form or two? Okay. So we need to know what its pKa is.
pKa would be 6.
Okay. That's one you definitely need to have memorized. That's one of the more important ones to have memorized. Okay. So is this the correct form at physiological pH?
Yes.
Okay. Now, physiological pH is about what?
7.4.
Good. Why don't you then, off to the right, draw what this would look like, say, at a pH of 5. Let's draw the histidine at a pH of 5. Let's draw a new picture for that one.
Okay.
Rather than, you can label that the first picture is at pH of 7.4, and then let's draw a new picture for pH of 5.
So, 2, 7.
actually. Okay, the same thing.
So should the side chain be protonated or deprotonated?
Protonated. I just don't know what to do with double bonds. Do I change them around?
Okay, so it looks like you're not quite sure how to draw the two different forms. Well, look at the neutral form off on the left. Which of those nitrogens is going to get protonated? Which of the two nitrogens is going to be protonated?
One on the right.
That's right. That's the one that's going to be protonated. Okay, so let's just add a proton to it. You don't have to do anything to the double bond because it's going to use its lone pair to protonate.
Okay.
Okay, something went wrong with the nitrogen at the top left corner now.
Yeah. Sorry.
You're writing it with a negative charge?
Would I just transfer the double bond?
Okay, no, I guess we're getting a little confused about that. So that nitrogen doesn't change. So generally speaking, in the side chains, only one atom gets protonated or deprotonated. I guess this is kind of the same confusion we had going on with the arginine. Only one atom is going to get protonated or deprotonated. I think maybe you were getting confused by table 3.1 because they're trying to draw resonance. They're trying to draw how the double bonds spread out in resonance. But that's probably not necessary for your course. So for our purposes, we can just say that in each acidic or basic side chain, only one atom is going to protonate or deprotonate. Okay, so make sure you have these two pictures in your notes. These are the two pictures of histidine. Again, maybe the pictures in table 3.1 aren't too useful to us here because the protonated picture uses resonance. So rather than using the picture in table 3.1, perhaps you should make sure you have these two pictures down in your notes as the correct pictures for histidine. This is a very important amino acid that's pretty likely to come up on the test, so it's very important to know the correct picture here. Okay. Since this will be for our notes, let's draw that double bond on the nitrogen a little bit neater so that you'll know what that is when you come back to it. Okay, so label the left-hand picture as pH of 7.4, please. And the right-hand picture you've got is pH of 5. Good. Okay. So let's see, where were we? So let's go back to physiological conditions. Under physiological conditions, what type of side chain does histidine have?
I would say polar.
Yeah. Is it positively charged?
No.
But here's where things are a little bit tricky. It's not. You said it's not positively charged because 6 is less than 7.4, right?
Yeah.
But that's true. But 6 is not that much less than 7.4. 6 is actually pretty close to 7.4. In fact, 6 is about 1 unit away from 7.4, right? If you rounded off the 7.4 to 7, you could say it's about 1 unit away. So because of that, even though the neutral form is the major form, there's still going to be a significant fraction of the protonated form as well, even at physiological pH. Even at physiological pH, even though the major form is neutral, there will still be a significant fraction of the protonated form because 6 is pretty close to 7.4. In fact, do you remember that exercise that we were doing earlier? It's actually maybe... So what's physiological? Let's go back to my screen. What's physiological pH?
7.4.
And what's the pKa for the histidine side chain?
About 12.5.
I think that was a mistake.
I'm sorry, 6.
Good. And actually, I'm going to round off physiological pH to just 7 here. So let's use the Henderson-Hausebach equation. What should I write down for the Henderson-Hausebach equation?
For the Henderson-Hausebach, pH is equal to pKa plus the log of the acid over the conjugate base over the acid.
Maybe in this case, should we call the top the protonated or the deprotonated form?
The deprotonated form.
So the bottom is the protonated form. So in this case, what would we plug in for the pH? About 7. And what do we plug in for the pKa?
6.
So let's try to solve for the ratio of deprotonated to protonated. How would we solve this equation for the ratio of deprotonated to protonated?
I'm not too sure.
Let's see. No, I think we've done questions like that before.
The ratio of protonated to deprotonated would be 1:1.
Okay, well, that's incorrect. I think we've done problems like that before. The first thing we have to do is get both the 7 and the 6 on the same side of the equation.
Okay. Oh. Okay, I thought you wanted me to get a numerical value for them. Sorry.
Eventually, we want a numerical value, but the first step is to simplify the equation. So
6, subtract 6 from 7.
So what would be the new equation?
1 is equal to the log of the deprotonated over the protonated.
All right, how could we continue to solve for the ratio then?
The ratio is 10 to the first is equal to deprotonated over protonated.
Excellent. That's exactly right. That's using the log skills that we went over before. So what does this tell us? Is it is there more deprotonated or more protonated? Or what is 10 to the first?
10 to the first is 10.
So is there more deprotonated or more protonated?
There is more deprotonated.
Which we already know from your screen. We already knew it was mainly in the deprotonated form. How many more times? How much more deprotonated than protonated?
10 more than protonated?
Yes. So would you agree approximately 10% is going to be protonated?
Yes.
Not exactly, but approximately. The ratio, for every 1 protonated, there will be 10 deprotonated. For every 10 deprotonated, there will be 1 protonated. So approximately about 10% will be in the protonated form. Okay, so this is a good application of the Henderson-Hausebach equation. You might have to do a problem like this on the test. I'm not sure if your instructor will test this. Many courses test this using the Henderson-Hausebach equation, this type of problem. In any case, we used some of the log skills we talked about. So here's my point. We know that at physiological pH, histidine is mainly in the deprotonated form, but there's still a significant 10% fraction that's protonated. What we learned from this is because 6 is only 1 unit less than 7, because 6 is less than 7, but it's only 1 unit less than 7, there's still going to be a significant amount of the minor form, about 10%. The ratio of one form to the other will be about 10 to the first to 1, 10 to 1. Okay. So what we just showed that is that because 6 is less than 7, most of the histidine in the body will be in this neutral form in the side chain. However, because 6 is only 1 unit less than 7, there will be a significant fraction, about 10%, that's in the positive form. This turns out to be very important. What this really means is that there's an equilibrium. There's an equilibrium between these two forms. Each histidine side chain is kind of constantly going back and forth. It spends some times looking like this, and some of its time looking like this. Which side of this equilibrium is favored at physiological pH, the left or the right?
The right.
Like so?
Hold on. No, the left.
Like so?
Yes.
Yeah, that was the whole point. Remember, about 90% of the molecules will be in this form and only 10% in this form. Another way to put that is each histidine spends about 90% of its time looking like this and about 10% of its time looking like this. About 10% of the time, the nitrogen will be protonated, and then it'll lose the proton and spend 90% of its time looking like this. But then 10% of the time again, it'll pick up a proton. So each histidine is constantly moving back between these two different forms, protonated and deprotonated. You might say it spends 90% of the time like this and 10% like this. And if you alter the environment, of the histidine, you can change how often it gets protonated or deprotonated. So histidine is a very important amino acid because it tends to take two different forms in proteins, which can be an important part of the protein's actions. So histidine is likely to be tested. All right, so this is a little bit complicated. So the important thing here is, even though the major form at physiological pH is neutral, this 10% positive is still very important. This is still a significant fraction. The fact that it can spend 10% of the time positive is important. And also, because this 6 is close to 7, if we make minor changes in the environment of the histidine, we could get more or less of it to be protonated. Here's these the two forms we were talking about. Histidine spends about 10% of its time in this form and about 90% in this form. So, which type of side chain does histidine have at physiological pH? Your textbook puts histidine in the positively charged side chain category. Your textbook puts histidine in the positively charged side chain category because this 10% turns out to play such an important role. So it's really kind of borderline. It's 90% neutral, 10% positive. What I was saying is your textbook actually puts histidine in the category of having a positively charged side chain, even though it's only charged 10% of the time. This 10% is so important that it actually puts it in the positively charged side chain category. But really, it doesn't fall into any of the categories neatly. It's a special case. It's kind of borderline. The main form is neutral, but this positive form is very important, even at physiological pH. This 10% is still a significant number. Okay, so like I said, that's important information about histidine that's likely to be tested. Is that making any sense?
Yes.
All right, the most important thing is just to make sure you can draw the two forms. The most important thing is to make sure you can draw the two forms. Let's go back to your screen. Okay. Now let's try drawing proline. Let's try drawing proline at physiological pH. Okay. Oh, you've already drawn it. Let's see. Point to the alpha carbon. Okay, that's excellent. All right, so it looks like you actually done a pretty good job of learning that structure. That's a tough structure for many students to draw, but you learned the correct way to draw that on your own. That's very good. That's what it looks like at the physiological pH. So obviously proline is unique because it has that ring structure. Proline is very interesting because it has that ring structure. One thing that's important about proline then, if there's does proline in a peptide, does proline increase the rigidity or flexibility of the peptide?
Rigidity.
Yes, because rings are rigid. That ring structure increases the rigidity of that part of the protein. That's an important aspect of proline that's important to keep in mind. How about glycine? Does glycine increase rigidity or flexibility for the peptide?
Glycine, flexibility,
Why?
because it's doesn't have a side chain.
Yeah, or it does have a side chain. What is the side chain?
2 hydrogens.
Well, one hydrogen. One of the hydrogens is just the normal part of the main chain. So the side chain is a hydrogen. That's the smallest possible side chain. Since the side chain is so small, there's very little steric hindrance around glycine, which increases flexibility. Having such small steric hindrance increases flexibility. That's often tested. So the more glycines a peptide has, the more likely it is to be flexible. And the more prolines it has, the prolines introduce rigidity into the molecule. Okay, so that's good that you had learned how to draw the proline. Good. Okay, so I can see you said when we started today that you had kind of memorized a lot of the amino acids, and I can see you have. A lot of those you made some good progress on. So one new thing here is you kind of also have to memorize table 3.1. We didn't talk about everything in the table, so there's some more amino acids in there that you should know, except remember the table tends to draw the 2 different forms using resonance, and it's probably better not to use resonance. That's kind of confusing. So the structures that are drawn with resonance, you should probably try to figure out how to draw them without resonance. But you should memorize which amino acids have 2 different forms and the ballpark pKa's from that table. Those are likely to be tested. You can see you can't figure out the correct. One of the types of problems that's likely to come up on the test is asking you to draw the charges on an amino acid at a certain pH. And you can't do that if you don't know the ballpark pKa. So those types of questions I've been giving you are pretty typical part of the first test for your biochem course. Okay. So like I said, I think you've done a pretty good job on memorizing a lot of the amino acid side chains, except maybe the ones that have 2 different forms. So make sure that you work now on making sure you have memorized the 2 different forms for the side chains that have 2 different forms. The sooner you get that down, the better, because your class is moving on to new material this week.